Hi everyone, welcome back to my channel. Let us know about the bacterial cell wall and peptidoglycan structure. Coming to the introduction of bacterial cell wall. The cell wall is the layer, usually fairly thin, that lies just outside the plasma membrane. It is one of the most important prokaryotic structures for several reasons. It helps determine the shape of the cell, it helps protect the cell from osmotic lysis. It can protect the cell from toxic substances, and in pathogens, it can contribute to pathogenicity. The importance of the cell wall is reflected in the fact that relatively few prokaryotes lack cell walls. The prokaryotic cell wall also is the site of action of several antibiotics. Therefore, it is important to understand its structure. After Christian Gram developed the Gram stain in 1884, it soon became evident that most bacteria could be divided into two major groups based on their response to the Gram stain procedure, that is, Gram positive bacteria and Gram negative bacteria. Gram positive bacteria stained purple, whereas Gram negative bacteria stained pink or red by the technique. The gram-positive cell wall consists of a single 20 to 80 nanometer thick homogeneous layer of peptidoglycan lying outside the plasma membrane. The gram-negative cell wall has a 2 to 7 nanometer peptidoglycan layer covered by a 7 to 8 nanometer thick outer membrane. Because of the thicker peptidoglycan layer, as the walls of gram-positive cells are more resistant to osmotic pressure than those of gram-negative bacteria. Microbiologists often call all the structures from the plasma membrane outward, that is, cell envelope. Therefore, this includes the plasma membrane, cell wall, and structures like capsules. One important feature of the cell envelope is a space that is frequently seen between the plasma membrane and the outer membrane in electron micrographs of gram-negative bacteria, and is sometimes observed between the plasma membrane and the wall in gram-positive bacteria. This space is called the periplasmic space. The substance that occupies the periplasmic space is the periplasm. The nature of the periplasmic space and periplasm differs in gram-positive and gram-negative bacteria. Coming to the peptidoglycan structure. Peptidoglycan or murin is an enormous mesh-like polymer composed of many identical subunits. The polymer contains two sugar derivatives, N-acetylglucosamine and N-acetylmuramic acid, and several different amino acids. Three of these amino acids are not found in proteins, that is, D-glutamic acids, D-alanine, and mesodiaminopimelic acid. The presence of D-amino acids protects against degradation by most peptidases, which recognize only diel isomers of amino acid residues. The peptidoglycan subunit present in most gram-negative bacteria. The backbone of this polymer is composed of alternating N-acetylglucosamine and N-acetylmuramic acid residues. A peptide chain of four alternating D and L amino acids is connected to the carboxyl group of N-acetylmuramic acid. Many bacteria replace mesodiaminopimelic acid with another diamino acid, usually L-lysine. In order to make a strong mesh-like polymer, chains of linked peptidoglycan subunit must be joined by cross-links between the peptides. Often, the carboxyl group of the terminal D-alanine is connected directly to the amino group of diaminopimelic acid.